Electrophoresis is a separation technique based on the movement of charged molecules in an electric field. The technique is used to separate proteins according to mass. In principle, electrophoresis of proteins works in the same way as electrophoresis of DNA. In practice, there are several crucial differences. Firstly, DNA base pairs are about 6 times heavier than amino acids. Therefore, most DNA fragments are larger and heavier than most proteins. Consequently, a finer gel matrix is required to separate proteins. Instead of the agarose gel used to separate DNA molecules, a polyacrylamide gel matrix is required for protein separation. Polyacrylamide gel electrophoresis is frequently referred to as PAGE. Secondly, whereas DNA is usually linear, most proteins are folded up into globular shapes that may comprise 2 or more subunits. To ensure that the folded shape does not affect movement through the gel, proteins are denatured before electrophoresis. Denaturation destroys the 3-dimensional structure of the protein and reduces multi-subunit proteins to a single linear chains of amino acids, the primary structure. Thirdly, whereas DNA has a uniform negative charge due to the phosphate groups, some amino acids are positively charged, others are negatively charged, and others still are neutral. Therefore, depending on their amino acid sequences, different proteins have different overall charges under physiological conditions. This is a problem because charge affects the direction and rate of movement during electrophoresis. We want all proteins to move in the same direction, and we want separation to be according to mass. We solve the requirement for denaturation and a uniform charge by addition of a negatively charged detergent, sodium dodecyl sulfate, SDS. This is the SDS in SDS-PAGE. In the presence of SDS, the proteins unfold, and SDS molecules bind indiscriminately to all proteins in a sample, masking the charges of the individual amino acids and giving the proteins a uniform charge-to-mass ratio. When an electric field is applied, all proteins in the sample have the same negative charge per unit of mass. All will migrate towards the anode and will separate on the basis of mass alone. To prepare protein samples for SDS-PAGE, we simply add a pre-prepared loading buffer. The loading buffer contains SDS to denature the proteins and give them a uniform negative charge, beta-mercaptoethanol to break cysteine-cysteine covalent bonds, glycerol to ensure that the samples sink to the bottom of the wells, bromophenol blue, a tracker dye that will indicate the progress of electrophoresis. The samples are boiled briefly to ensure complete denaturation. The agarose gels used for electrophoresis of DNA are quite thick, about 1 centimeter, and are mounted horizontally. The polyacrylamide gels used for electrophoresis of proteins are thin, about 1 millimeter, and are usually mounted vertically. A polyacrylamide gel is actually 2 gels in one, a stacking gel in which each protein sample is concentrated into a single narrow band. And a separating gel, where the proteins separate out according to mass. A polyacrylamide gel mold is made as follows. A U-shaped rubber gasket is placed on top of the notched plate, outside and around the 2 raised ridges, called spacers. The ridge on the gasket must be facing towards you, just as the ridges on the plate are facing towards you. A plain rectangular glass plate, called the back plate, is placed on top, and the plates are clamped together to form a mold. The separating gel is now poured in to about 2/3 full of the mold. Butanol saturated water is layered over the liquid gel to keep the top of the gel flat and to exclude oxygen, which inhibits polymerization. The composition of the separating gel is acrylamide, a small organic molecule capable of polymerization. Warning alert here, liquid acrylamide is highly toxic. It is less dangerous when in the polymerized gel form, but it should still only be handled when wearing gloves. Bisacrylamide, which cross-links the polyacrylamide chains. SDS, a high ionic strength tris chloride buffer, pH 8.8, and polymerization reagents, TEMED and ammonium persulfate. When the gel is polymerized, i.e. set, the butanol saturated water is washed away, and the mold is filled to the brim with stacking gel. A plastic comb, shown here in red, is forced between the plates into the liquid stacking gel to create wells. The stacking gel has the same components as the separating gel except a lower concentration of acrylamide, hence a more open matrix, neutral tris chloride buffer rather than the basic buffer used in the separating gel. The difference in pore size and pH between the stacking gel and the separating gel lead to the stacking effect, whereby each protein sample is concentrated into a single narrow band, so that all proteins enter the separating gel simultaneously. When the stacking gel is fully set, the clamps, rubber gasket and comb are removed. The gel is submerged completely in an electrophoresis tank containing a high ionic strength tris glycine buffer. Most electrophoresis tanks can run 2 gels at a time back to back, but only one is shown here for simplicity. Samples are added to the wells by pipette. One well is loaded with a mixture of proteins of known molecular mass, the standards, or protein ladder. Note that once the gel is in place, the buffer at the cathode is separated from the buffer at the anode so that the electric current must pass through the gel. The tank is covered with a lid and connected to a power pack. When the electric current is switched on, negatively charged ions travel from the cathode through the buffer to the anode through the gel, and proteins migrate downwards through the gel matrix. Never touch a liquid while there is an electric current running through it. The proteins are not visible to the naked eye, but the blue dye in the loading buffer, bromophenol blue, can be seen migrating through the gel. The dye, being of low molecular mass, runs faster than the proteins. When the dye approaches the end of the gel, the current is switched off, and the gel is removed from the plates. The most common visualization method is Kamasi blue stain. The gel is soaked in Kamasi blue dye and then de-stained with solvents. The blue dye binds indiscriminately to all proteins on the gel. By comparing the bands to the protein ladder on our left, it is possible to estimate the molecular mass of the various proteins. Smaller proteins will have migrated farther through the gel matrix, so they will be towards the bottom. Sometimes we may wish to identify a specific protein on the gel. Instead of Kamasi staining, we perform what is called a Western blot. The unstained gel is placed in contact with a membrane. In the arrangement, the membrane is connected to the anode. When an electric current is passed through the gel, the proteins move to the membrane. The membrane is then probed with an antibody against the specific protein of interest. Let's call it an antibody against protein A. Here in the result, the ladder is on the left and the three bands towards the right side of the gel have the same mass and have all cross-reacted with the antibody, so all three must be samples of protein A. SDS-PAGE followed by Western blotting is one of the most commonly used techniques in a molecular biology lab. Take-home points. SDS-PAGE is a type of electrophoresis in which proteins are separated according to mass. SDS detergent gives the proteins a negative charge, so they migrate towards the positive electrode, the anode. Smaller proteins pass through the gel more easily, so they migrate faster and further, i.e., distance moved is inversely proportional to size. After staining, the larger proteins are visible as bands near the top of the gel, whereas smaller proteins will be near the bottom. Comparison with the molecular mass standards, the protein ladder, reveals the approximate size of the various proteins.